Welcome everyone. Can you all hear me? Okay, great. So we have about 4 yards of DNA in each of our cells, and if you put it all back together, back to back, you can go around the Earth 100,000 times, to the moon and back about 5,000 times, and to the sun and back 10 times. So I think this is just an amazing field, and I hope to instill some of that enthusiasm in you as we progress through our talks here. So I'm going to give you an overview first of the basic principles and types of mutations that we encounter in the molecular lab, the detection of specific known mutations, the method's pros and cons, and I will provide lists of those in the context of disease examples, and then we'll do some interactive results interpretation as well. Don't worry, I won't put you on the spot, but if you want to interact, then please do. And we'll also talk about sequencing and about mutation screening methods. So first, let's go to the basic principles and types of mutation. For many of you, this will just be a refresher. But you'd be surprised when I ask people what constitutes a gene, the diversity of answers that I receive. So what constitutes a gene? Well, as you know, by convention, we have 5' to 3', and that basically is because the carbon on the 5' end is not linked to a sugar group on the other end. And there are phosphodiester bonds, 5' to 3', from the polynucleotide chain to the adjacent deoxyribose groups. And then in the gene itself, we have a promoter, where basically transcription factors bind and activate the gene. Then we have the start of transcription, which is the messenger RNA transcription. And you can see that this already occurs in an exon. So it's not true that all exons are completely protein coding. Then there is the 5' untranslated region, the initiation of translation, which is called the initiator codon, and that's always a methionine. And then in blue we have the exons, and the bars represent the introns, or the intervening sequences. And then at the end there is the termination codon, then again a 3' untranslated region, which again is messenger RNA but not transcribed into protein, or translated into protein. And then we have the polyadenylation signal. So those three are the building blocks of a gene. Now when we look at DNA mutations, that's any change in the consensus sequence of a gene. And there is an average DNA mutation rate. Most of those are ultimately repaired, but some of them are not. And especially in mutation hotspots, throughout the genome, we see an increased number of mutations. Now, mutations can be polymorphisms, and that requires some definition. A mutation is something that has an allele frequency less than 1%, whereas something that is a polymorphism has an allele frequency greater than 1%. And it means that some polymorphisms, some common variants, can certainly contribute to disease or cause disease, whereas some rare variants, which theoretically would fall into the category of mutation, do actually not cause disease. But colloquially, we use mutation for something that is disease-causing and polymorphism for something that is more benign. But it's better to basically talk about sequence variants. Polymorphic sites can be useful in diagnostic testing with restriction fragment length polymorphisms and variable numbers of tandem repeats. And mutations can also be silent.